Yeah, hello, welcome to this video lesson. This is biology form 3, Bellington Kamisa. I'll take you through translocation. As you can see, the picture is really depicting what actually translocation is all about. You can see source of the cell leaf, the companion cell, this one, and the substances, which is sucrose moving from there. We can also see water moving within. So you can suggest, you can define from there. Uh, um, Let us understand the definition of translocation in the next slide. Here we are going to look at the specific objectives. By the end of this lesson, you should be able to define the term translocation. You have to name the substances that are translocated in plants. You have to define the term diffusion. You have to explain the factors that affect the rate of diffusion. Now here we are, translocation. As we mentioned earlier on, the phloem translocates manufactured food substances from the leaf cells and the storage tissues to all other parts of the plant. So this is by a process called translocation. So therefore, we are saying the food substances move from the leaf cells to the sieve tubes by active transport. So active transport is the movement of food from the leaf cells to the sieve tubes against concentration gradient. So we are saying the energy for this process is provided or is produced by the mitochondria, which is located in the companion cells. So the companion cells are actually supplying this energy to the leaf so that the food can move from leaves to the sieve tubes by active transport. Now how translocation takes place. So we are saying manufactured food substrates move from the leaf cells to the sieve tubes by active transport. So we are also saying companion cells in the phloem provide energy for the process of active transport. So as the substances accumulate in the sieve tubes of the phloem in the veins of the leaves, water is drawn by osmosis from the adjacent cells. Here, adjacent cells, we are trying to mean the neighboring cells. Right? The entrance of water into the sieve tubes increases hydrostatic pressure in the tubes. So this results in movement of the manufactured food substrates from one sieve tube to another through the sieve pores. Now the food molecules stream over the cytoplasmic filaments in a process called cytoplasmic streaming from one sieve tube to another or to the next. This process uses energy provided by the companion cells as well. Now, in order to carry out the life processes that we mentioned previously, a cell needs and takes in various substances. So in carrying out these processes, the cell produces certain substances as well. So some of these are waste products, while others are substances that the cell does not need but are useful to other cells in the tissue. So such useful substances are usually taken out of the cell to other cells that need them. So therefore, substances are always moving into and out of the cell. In most cases, substances move into and out of the cell depending on what the cells require. So the way substances move into and out of the cell depends on certain properties of the substances, such as size of the molecules and the type of the substance. So in most cases, large molecules may not pass through the cell membrane. So there are three main physiological processes by which substances move into the cell and out of the cell. So these physiological processes will have diffusion, osmosis, as well as active transport. So in this section, you will learn about the three processes the factors that affect them and their importance in plant and animal tissues. But basically, in this lesson, we will dwell very much on, will center much on diffusion. Let us look at diffusion as a physiological process. So we are saying diffusion is demonstrated when a drop of ink is placed into a glass of water. The ink particles seem to spread in the water until all the water is uniformly colored. So you are also able to smell perfume that other people have worn because the particles of perfume diffuse from them through air to our organs of smell, that is the nose. So generally here, if a lady maybe is entering the door, when he steps the leg on the at the door, you would smell the perfume. That is by simple diffusion. That's why you are able to smell the perfume. So in order to study the process of diffusion, one can carry out the following activity so that he or she understands how diffusion takes place. You know, diffusion takes place in two states of matter, liquids and gases, but not in solids. Right? This activity will be done in order to understand, to demonstrate the physiological process of diffusion. So to demonstrate diffusion using the Visking tubing, one would need these materials: Visking tubing itself, starch solution, dilute iodine solution, beakers, string. The strings are used for tying the Visking tube. Here, here we are, the procedure, which is the steps that are followed. One is there to measure and cut the Visking tubing 8 centimeters in length. Then you can tie one end tightly with a string about 1 centimeter from the tip. We'll fill the Visking tubing with starch solution and then tie the other end. Then you have to note the color change of the solution. Then you have to dip this Visking tubing into a beaker filled with dilute iodine solution. You leave it to stand for 15 minutes. The fifth one, you have to make observations on the color changes and complete the table below. So here we have the xylem before, after 15 minutes, then the conclusion. Then you observe the color of the starch solution inside the Visking tubing before the experiment, and after the experiment, you do the conclusion. Likewise, the color of iodine solution in the beaker before the experiment and after 15 minutes, and then you conclude. So to understand how you can fill in here the table, we have to go through the next slide. Now, questions, you'll be guided by these questions to understand how these procedures are conducted. So you need to find this. Why did the color of starch solution change after the Visking tubing was mixed in a solution of iodine? And again, you need to give the reason. And number two, why was there no change in the color of iodine solution in the beaker after 15 minutes? The third one, the Visking tubing has some molecules to pass through and not others. What is the name given to the membrane that has this character of actually selecting others and leaving out others? Right, so you observe that the color of the starch solution changed from basically from brown to blue-black because iodine molecules moved into the Visking tubing, which brought about the change. So the color of iodine remained brown because starch molecules did not move from the Visking tube to the beaker because they are large molecules. The membrane is permeable to iodine solution but not to starch because starch molecules are large molecules. So now here with me I have a video which will be played to cement the understanding, to demonstrate how diffusion takes place. And here, with me, you watch this video.
This is visking tubing. It's long been used in school biology practicals. It's semi-permeable, which makes it useful as a model of the human gut, which is what I'm going to show you in this demonstration. I'm going to fill a piece of tubing with a mixture of starch and glucose, and I should be able to show that over time the smaller glucose molecules migrate through it, whereas the larger starch molecules do not. So we need to soak the visking tubing in water for a few minutes, then it should be flexible enough to work with. You want to hold one end of the tubing open, and an easy way to do that is to use the end of a saw.
Thank you. You can continue enjoying this video. This is the video which is explaining another one way of doing the same experiment to demonstrate how diffusion occurs using the visking tubing. That's what you are supposed to do. Right, diffusion is the movement of particles of substance from a region of high concentration to a region of low concentration. So we are saying the difference in the concentration of the particles in the two regions is called a concentration gradient or diffusion gradient. Sometimes the concentration gradient is also called the diffusion gradient. Now, diffusion occurs in liquids and gases and does not take place in solids. Yeah, diffusion of certain particles also takes place through the cell membrane, cell walls, and cell cytoplasm. So when particles move from a region of high concentration to a region of low concentration, they are said to diffuse along a concentration gradient. So as long as a concentration gradient is maintained, the movement of the particles continue until they are evenly distributed or spread in the available space. Now let us, having looked at, demonstrated how diffusion occurs, we have to look at the factors that affect the rate of diffusion when it is taking place. So the rate, what is the rate now of diffusion? The rate of diffusion of a particle is the time taken for the particles to diffuse within an available fixed space until they are evenly distributed. So there are several factors that affect the rate of diffusion, and these factors include, so highlight them in the next slide. So temperature is one of the factors that actually affect the rate of diffusion. Now, when the temperature of the particles increased, their kinetic energy also increases and the particles move faster. Therefore, we are saying the higher the temperature, the faster the particles will diffuse, and the lower the temperature, the lower the rate of diffusion. You know, from the kinetic theory of matter, we learned that matter contains the particles. When these particles gain energy, they vibrate more and collide. They move at Brownian motion. So you can see that one. Another factor is concentration of the substance. Now, a greater difference in the concentration of the particles between two regions results in steeper concentration gradient and vice versa. So therefore, diffusion is faster when the concentration gradient is very high. So the difference in concentration of the substances determines the rate of diffusion, how fast the particles will move. So where the difference in concentration of the substance is big, the rate of diffusion is high because this increases the concentration gradient or slope. Now we have also the size of the particles here. Small particles diffuse faster than large particles, and we have this is because the small particles are lighter and pass through air or water easily. So we have another factor which is thickness of the membranes and tissues. Now thin membranes enhance higher rate of diffusion than thick membranes. So this will shorten the distance that the particles automatically diffuses. Right, so we have another factor which is surface area to volume ratio. It's another factor which affects the rate of diffusion. Now, the higher the ratio, the greater the rate of diffusion. So lower organisms have got a higher surface area to volume ratio, and that one increases diffusion. If there is high surface area compared to volume ratio, the rate of diffusion increases. So now distance a particle has to move, an even distribution of particles is reached faster when the distance involved in diffusion is small compared to larger distance. Now, let us look at the significance of diffusion. Why diffusion is vital? Why is it important to simple living things and as well as to large organisms? So we are saying it is involved in movement of gases into and out of the plant tissues. Remember, during the day, the carbon dioxide from the atmosphere is high than into the leaf. So therefore, the carbon dioxide diffuses from the leaf, from the atmosphere into the leaf cells through the stomates. So at the same time, once photosynthesis has taken place, the oxygen is produced and then diffuses out of the leaf, since it is at higher concentration than what is in the atmosphere. So therefore, we are saying oxygen therefore moves by diffusion from the leaf to the atmosphere. That's why diffusion is very, very important. It is able to make sure that the organisms meet their demands in their bodies. The second one is helping evaporation of water from the plant during transpiration. Water vapor is at higher concentration in the air spaces of the leaf than in the atmosphere. So the water vapor moves by diffusion from the air spaces to the atmosphere. That marks the end of a lesson, and here with me I have the review exercise to actually cement your understanding. You have to state the types of vesicular tissues. Number 2, you have to state the types of xylem tissues. Number 3, you have to name 2 cells that form the phloem tissue. Number 4, give the difference between the structure of the xylem and the structure of the phloem. Number 5, you have to define what is translocation. Number 6, you have which vascular tissue does the following functions. Numero 1, transports water. Numero 2, transports mineral salts. Numero 3, transports glucose. Number 7, we have to discuss any 5 factors that affect the rate of diffusion. We have to write our responses in essay form. Yeah, for any questions, you WhatsApp this number, help@xulendo.organization. You visit Xulendo online, xulendo.organization. For resources, references, we have taken some of the text from Cerebral 2017, Excel and Succeed, Biology, Book 3. The other one is Shimocha Similon, 2017, Achievers Senior Secondary Biology Student, Book 3. For illustrations, design, and graphics, you have to this is done by Charlotte Bruning and Dave Mumba on work Xulendo. Photos, we have taken them from this link. Thank you very much for being such a wonderful audience. I hope you promise to see you in the next lesson where we'll be concentrating on the other physiological processes, osmosis, as well as the active transport. That's what will be done in the next lesson. Stay well.